The relationship between bioelectricity and cancer is an emerging area of research that explores how electrical signals in cells and tissues influence gene expression, cell behavior, and morphogenesis. Defective bioelectricity during development, morphogenesis, could indeed play a role in activating pro-cancer genes or disrupting normal tissue organization, potentially contributing to cancer initiation and progression. Here's an overview of the latest developments in this field. 1. Bioelectricity and morphogenesis. Bioelectric signals. Cells maintain a resting membrane potential, voltage difference across the cell membrane, that influences ion fluxes, signaling pathways, and gene expression. Morphogenesis. During embryonic development, bioelectric patterns guide tissue organization, organ placement, and cell differentiation. Experiments, such as those by Michael Levin and colleagues, have shown that manipulating bioelectric signals can induce organ growth in specific locations or even regenerate tissues. Cancer connection. Disruptions in bioelectric signaling during development or in adult tissues could lead to misregulation of cell proliferation, migration, and differentiation, hallmarks of cancer. 2. Defective bioelectricity and cancer. Altered membrane potentials. Cancer cells often exhibit depolarized membrane potentials compared to healthy cells. This change in bioelectricity can activate oncogenic pathways and suppress tumor suppressor genes. Ion channels and transporters. Dysregulation of ion channels, e.g., potassium, sodium, calcium channels, and transporters can alter bioelectric signaling, promoting cancer cell survival, invasion, and metastasis. Bioelectric microenvironments. Tumors create unique bioelectric microenvironments that support their growth and evade immune detection. For example, cancer cells can manipulate extracellular pH and ion concentrations to their advantage. 3. Latest developments in bioelectricity and cancer. Recent research has focused on understanding and targeting bioelectric mechanisms in cancer. Here are some key developments. A. Bioelectricity as a diagnostic tool. Researchers are exploring the use of bioelectric signatures, e.g., membrane potential, ion channel activity, as biomarkers for early cancer detection and prognosis. B. Targeting ion channels for therapy. Ion channels and transporters are being investigated as therapeutic targets. For example, potassium channels. Blocking specific potassium channels can inhibit cancer cell proliferation and induce apoptosis. Sodium channels. Sodium channel inhibitors are being tested for their ability to reduce cancer cell invasion and metastasis. Calcium signaling. Modulating calcium signaling pathways can disrupt cancer cell survival and communication. C. Bioelectric reprogramming. Scientists are experimenting with bioelectric reprogramming to normalize cancer cells. For instance, restoring normal membrane potentials in cancer cells can reduce their malignant behavior. Using optogenetics, light-controlled ion channels, to manipulate bioelectric signals in tumors. D. Bioelectricity and the tumor microenvironment. The bioelectric properties of the tumor microenvironment, e.g., extracellular matrix, immune cells, are being studied to understand how they influence cancer progression and response to therapy. For example, altering the bioelectric microenvironment can enhance the effectiveness of immunotherapy. E. Bioelectricity in cancer prevention. Research suggests that maintaining proper bioelectric signaling during development and in adult tissues could reduce the risk of cancer. This includes ensuring proper ion channel function and membrane potential regulation. 4. Key experiments and findings. Michael Levin's work. Levin's lab has demonstrated that bioelectric signals can control large-scale patterning and organ regeneration. For example, they have induced the growth of eyes in unusual locations in tadpoles by manipulating bioelectric gradients. Cancer cell reprogramming. Experiments have shown that restoring normal bioelectric patterns in cancer cells can reduce their tumorigenic potential, suggesting that bioelectricity could be a new avenue for cancer therapy. 5. Challenges and future directions. Complexity. Bioelectric signaling is highly complex and involves interactions between multiple ion channels, transporters, and signaling pathways. Therapeutic targeting. Developing drugs that specifically target cancer-related bioelectric changes without affecting normal cells is a significant challenge. Integration with other therapies. Combining bioelectric-based therapies with existing treatments, e.g., chemotherapy, immunotherapy, requires further research. Conclusion. Defective bioelectricity during morphogenesis and in adult tissues could indeed be a factor in activating pro-cancer genes or disrupting normal tissue organization. The latest developments in bioelectricity and cancer research highlight its potential as a diagnostic tool, therapeutic target, and preventive strategy. While the field is still in its early stages, it offers exciting possibilities for understanding and treating cancer in entirely new ways.